So, continuing on to the topic of application of basic plant tissue culture technique, which have been utilized for crop improvement programs. In my last lecture, I explained to you that how protoplast uh, technology, when developed, helped in testing concepts of possibility of two different genome coexisting together in a cell, and hybrids were generated by fusing two protoplasts from two different species, two different genera, and two different tribes. So it gave rise to a possibility that two different genome or genes can coexist in a cell effectively, and the genes express themselves and modify the trait for which a gene is responsible. I mean, of course, that was demonstrated later, but then the experiments like what I discussed in my previous session uh, clearly make a way where it could be possible for genetically transform a cell and regenerating a transgenic plant. In this session, I'm going to discuss with you another aspect which is associated with in vitro plant regeneration in cultures, and the genetic variability which exists in a cell, starting cell, or it achieves while in culture, you know. So that genetic variability which it acquires during culture process, sometimes it is for good, and if that variability can be selected and tested, can give rise to an improved plant variety. So I'm going to look at that what are these somaclonal variants, how these variations are achieved, and how they get stabilized, and what are the limitations, etc., etc. Okay, so this is the slide I put every time, which has listed the application of tissue culture techniques, and I'm going to talk about the somaclonal variations. So let us look at what are somaclonal variations. Okay. So as a definition, if you have to define somaclonal variations, so these are genetic variations in plants that have been produced by tissue culture and can be detected as genetic or phenotypic trait. Okay, so basically in somaclonal variation, we are talking about genetic variation in plants which have been produced by plant tissue culture, which can be detected as genetic or phenotypic traits. And if it happens to be a useful trait, we can make use of that for improving, for seeing the improvement in that particular trait. Okay, so uh, what are the basic features of somaclonal variations? Okay, so it can exist in different levels. For example, variation for karyotype, isozyme characteristics, and morphology in somaclones may be absorbed. Okay. Now, it can be in Kelly clones, means the clones, you see, as I told you, the different, after inducing callousing, can be cut into small pieces, which represents a clone of callus, or mericlone, clone of meristem, or protoclone, and clones of protoplasts. So different, so what we are talking here is the genetic variability can exist in callus clones, meristem clones, or clones of protoplast. So clones is it's a group of cells, you know, and then generally heritable mutations and they persist in plant population even after plantation into the field. So this variability, which is seen at the clone level, calli clone, meristem clone, or protoclone, it is persisted, I mean, through the plant populations, even after plantation, and it goes to the field. So it means the whatever variation has taken place is sort of stable, which goes through these phases of plant development in vitro. Uh, so what is the mechanism of the somaclonal variation? Okay, so one is genetic, which is heritable variations. So it can be pre-existing variation in the somatic cells of X plant. Means the X plant which you are taking, in those somatic cells, there is some genetic variation which is pre-existing. That can be one source, or it is caused by mutation or other DNA changes in the somatic cell level. And these kind of changes, they occur at high frequency. What kind of? That is the heritable variations which exist in somatic cells, and also the changes which is caused by mutations and other DNA changes. Okay. Then second source is the epigenetic or non-heritable variations. And these non-heritable variations are created during tissue culture technique. And they cause a temporary phenotypic change. Meaning the genetic variation caused in the somatic cells during tissue culture, they are responsible for temporary phenotypic changes, and they occur at a low frequency. So we are talking about two mechanisms. One is genetic, which occurs at high frequency, and then you have non-heritable or epigenetic, which occurs at a lower frequency. So what is happening here? So you have these clones, callus clones, protoclones, or Kelly clones. Then organogenesis. You have regenerated plants. This is hardening process in the growth chambers, and then in the greenhouse, and then the somoclonal variations, variants you can select at this level for various phenotypic traits, if any. And that particular plant can be further reproduced or multiplied by sexual means, and the whatever variation one has detected at this level can be multiplied many times. So that these are the steps involved. So it is, we are not creating anything. It is either the genetic heritable changes or epigenetic changes which are occurring. Only thing is we are trying to exploit that change for our benefit. If it has happened for good, we just take that plant and multiply. You know. But then selection of that variation is very, very important. That we should be able to select that variation. Now, what are the causes? You know, so it can be a physiological change, cause, genetic cause, or biochemical cause, which are responsible for somaclonal variations. So, and let us look at one by one. So what are the physiological causes which can generate the somaclonal variations? The uh, one of the very important factor which causes this kind of variation is the exposure of culture to plant growth regulators. Now, these plant growth regulators can cause genetic change. Okay, and then the culture conditions also. So longer you keep a culture in vitro, in with growth regulators, more are the chances for getting genetic variability or losing the genetic uniformity. So that is one of the negative aspect of tissue culture. But then the occurrence of genetic variability during this was exploited for the benefit of crop improvement. As I mentioned, if some change has taken place, which is good, can be utilized for crop improvement program. Okay. Coming to the genetic cause, so one of the very basic change which is seen in tissue culture growth plant is the chromosome number. In the culture conditions, the chromosome number undergoes a change. Either it reduces or it increases the chromosome number in the tissue culture regenerated plants. So you have euploidy, change of chromosome sets, means diploid becomes tetraploid or polyploid, or you aneuploids means the loss or gain of a single chromosome or two chromosomes or three chromosomes. So that kind of genetic change does take place, Then the the chromosome structure can be changed because of these processes: deletion, inversion, duplication, translocation. So when the cell multiplies in presence of growth regulators, these kind of changes are seen. And these changes will occur either some part of the chromosome is deleted or inverted or duplicated or translocated from one region to another region. They result into the genetic variability and affecting sometimes the trait of the regenerative plant. So this is another cause, basically. Mm, Gene mutations can take place, uh, and then the transposable elements can get activated. Now we know that all genomes in all organisms, living organisms, has this presence of transposable elements which remain in their place. But once they move from one place to another, they change the genetic uh, makeup. As you know, the insertion of transposable elements can lead to mutations. It can be good mutation, it can be bad mutation. If it is good mutation, you can see if it is a good mutation, you can go for stabilization of that mutation and select that plant and further propagate. Uh, Another cause is the DNA sequence. It can change the DNA, it can change the protein, the DNA undergo methylation. All these processes I have explained to you in my preliminary lectures, that how a change in sequence can change the gene quality, and it can affect the proteins, and how methylation affects the gene expression, basically. Okay, so but they can happen in somatic cells, and when they happen, as you know, it leads to the very genetic variability. So when you have different clones and one clone, these things are happening, one not all together in one cell, but either this or this or this or this, they lead to genetic change. And then when it comes to biochemical causes, one is genetic, I told you, physiological we have discussed, not the biochemical causes can. I mean, the cell can undergo a change in its ability to photosynthesize due to change in carbon metabolism, or some alterations taking place. So that can lead to or affect the photosynthetic ability of a plant, you know, and then biosynthesis of starch via carotenoid pathway that can undergo a change, nitrogen metabolism, antibiotic resistance. So these are biochemical changes, biochemical causes which can occur or are responsible for somaclonal variations. Means a clone can show different kinds of photosynthetic ability, different kind of carbohydrate synthetic ability, nitrogen metabolism, and antibiotic resistance. You know, so and that change is because of the alteration into these pathways. You know? So that is because of the biochemical basis. Now, how these variations are detected? So I'm referring to when you have a plant regenerated through callus, protoplast, single cell, and then some kind of variation is seen, and that can be caused by physiological ways, genetic ways, or biochemical ways. The next question is how you detect them. Okay, so one is very simple through morphological characters. So if in the you have a population of tissue culture regenerated plant, and if you see that they are showing differences in their structure, in their biomass, and other things. So you can select those plants, you know, and then you can do some cytological detection because chromosomal aberrations can be seen through under microscope through cytological means. So you can examine that and see whether this change, which one is seen, is because of any change at the chromosomal level, you know, and then the DNA contents, you know, whether certain loss of DNA has taken place or gain of DNA in terms of when you have polyploids or aneuploids, that will be directly reflected onto the DNA content. So that is another way by which you can detect that any change is taking place by looking at the DNA content. You can check it on electrophoresis method by separating these macromolecules on gel and then look whether you see any difference among the different clones which you have. And then the traits. If a trait is, for example, disease resistance, so you can expose these plants to the pathogens and see which one is protecting better itself, you know. And then the herbicide resistance is another one. You can expose the population to herbicides and see which one is protecting itself. So there are different ways by which we can detect this somaclonal variations in a population of tissue culture regenerated plants. Okay. And I mean, in continuing with that, the stress resistance, you can expose the plants to different environmental factors of high salt, drought, flooding, you know, and then see if they are showing any variation in that. In that, the population is showing any variation in the stress tolerance. You know? So once you determine that, okay, out of 100 tissue culture regenerated plant, one plant or two plants are performing better. Then you go further with those plants and study what are the causes of that. As I mentioned, you can do variety of analysis that, okay, that is because of more DNA content, less DNA content, or like that, or any metabolic process undergone change and things like that. You know, so once you have identified, then it means that change which you are seeing in a trait is because of somaclonal variation. That the change which the somatic cell has undergone by any means which we discussed before. And these are the advantages, as I mentioned, of somaclonal variations, that it is directly expected to help in crop improvement. You know, you regenerate plants in vitro, large numbers, and then select those populations for different variants. And if it happened to be the one which you are looking for in terms of disease resistance or abiotic stress resistance or photosynthetic abilities or whatever, then you are done. So you have a plant which is better than the other, and it is based on tissue culture selection, okay, or somaclonal variants, not the genetic one, okay. And so there are so many advantages by which you can use this variation, which is caused by in vitro technique actually. I mean, during the what are the disadvantages of those? You know, the one of the disadvantages is that whatever clone you are starting with, you know, callus, protopore, protopore, calli, all these, they do not have the uniform, all the cells having the same change. So uniformity or uniformity as well as the genetic change is concerned is the biggest question here. And then sometimes you have the desired and also the undesirable trait coming into. So it's a random process, and you may have undesirable changes also taking place, you know, and and then it requires very extensive field trials, very laborious, you know, and then it is seen experimentally that this approach is not good for uh, complex traits of crops, you know, which are agronomically important traits like yield and quality of yield, you know, because these are very complex traits. It's not simple depending on single factors. There are so many factors which influences the plant productivity in terms of their quality and quantity. So if I have to, if I have to actually summarize what I have been talking about is that although a lot of potential was shown in this approach of crop improvement, looking at somaclonal variations. A lot of successful papers were published, a lot of literature is available of 70s and 80s, but then this could not get stabilized as one of the major ways of crop improvement. And because of the inherent problems I discussed with you, that the trait is not uniform in the plant because of the original the clone which one is taking is a group of cells where you may have the somaclonal variants and non-variants, you know. So again, it has just been there as one of the possibilities by which the tissue culture technique has been exploited for crop improvement. So with this, I conclude my sessions with you on plant tissue culture and its application for crop improvement. You know. Now, I will, in the forthcoming session, I will talk about uh, the the very interesting phenomenon of genetic transformation and gene transfer and raising transgenic plants. First, we will see how, you know, I have talked so far the developments took place up to late 70s and early 80s of last century. And they gave us all this information, background information. And some of the approaches have definitely contributed for crop improvement, like haploid production I mentioned. That is one. But all other things were there, and they were ready to find out the methods by which successfully foreign DNA can be transferred to the cell and transgenics can be produced. And that's one part of it, so that the modification can be directed. So we know that we want to change this trait, and this trait is guided by this gene or set of genes, and that if we push it here, regenerative plant, then this plant is expected to have a modified trait. So in next session, I will talk about how it was possible to genetically transform plant cells, because by the end of 70s or early 80s, it was realized those who were working on genetic engineering area, they almost gave up because those efforts were not bearing fruits. That is, to genetically modify a single cell or a chloroplast to raise transgenics. But then suddenly something happened in two diverse areas. One in the bacterial side, where the antibiotic resistance genes were discovered, and second, the understanding the the molecular basis of a plant disease we call ground ball disease of plants. So I will discuss about all this in my next session. Thank you very much.